Thank you very much. Thank you all for being here. It's the morning after the night before. You've all done very well. I might just pour myself a water because I danced and sang a bit too much yesterday evening. Thank you to the organisers for inviting me to speak, though I'm not so sure about this morning's session, but hopefully I'll be able to excite you and fill you with interest about the impact of developmental disorder-associated variants in the general population. So I have no conflicts of interest to declare. I'm a research scientist at the University of Exeter. So the key concepts I want to try and cover in this lecture are a brief introduction to monogenic developmental disorders. I'm going to touch on what we mean by incomplete penetrance and variable expressivity. I'm then going to talk about developmental disorders in the general population and some of the work that we and others have done looking at the sorts of phenotypes that are associated with these variants in population cohorts. And then I'm going to touch on the ascertainment biases between clinical and population cohorts and some of the reasons why we might see different penetrance in these different cohorts. And then finally, I'm going to talk, because this is an educational session in particular, I'm going to talk about some of the problems and challenges of doing these sorts of studies and where they can go wrong. So, monogenic developmental disorders. These affect maybe 2 to 5% of children, and they can be syndromic or non-syndromic. So there's a range of phenotypes associated with developmental disorders, intellectual disability being the most common, and developmental delay, but also things like seizures, congenital malformations, cardiac defects. And some individuals have multiple of those syndromes affecting multiple organs. They are by and large caused by large effect, single rare variants, and over 2,000 genes have been associated with developmental disorders at this point. And they essentially obey standard Mendelian patterns of inheritance, so autosomal recessive, autosomal dominant, X-linked. And most of today's talk I'll focus on autosomal dominant. And in these cases, of course, the presence of a causative genotype is highly predictive of disease. So we use genetic testing in the clinic to diagnose these individuals and to test their families for predictive testing and counselling for the recurrence risk and obviously to target specific treatments. And increasingly we're using genome-wide testing because, as I said, more than 2,000 genes are associated with these conditions. Many are phenotypically quite similar, and so rather than guessing which gene might be the cause, we do a genome-wide or whole genome sequence test. And we've known for a long time that these conditions are both genetically and phenotypically heterogeneous. So this plot shows you something of the genetic heterogeneity, where in the corner we've got cystic fibrosis, a condition where essentially just variance in one gene causes particular phenotypes. And as we go up the scale, you'll see that as the phenotype perhaps becomes more non-specific, more and more genes essentially produce phenocopies. So similar phenotypes can be produced by different variants in different genes, which obviously makes their diagnosis quite challenging. As I say, they can also be phenotypically heterogeneous, and I just wanted to give you one example of this from the Deciphering Developmental Disorders study. This is a UK-wide study to use genome-wide technologies to diagnose severe previously undiagnosed developmental disorders. I think we've been talking about it at SHG for about 10 years now, but we're still working on it. We've got over 13,000 patients involved in the study. And this particular example is KBG syndrome. This is our highest hitting diagnostic gene. We have nearly 80 patients with a diagnosis in ANCAR D11. And KBG syndrome is caused by ANCAR D11 haploinsufficiency. And what you can see in this graph is individuals in the DDD study with a loss-of-function variant in this gene. And a histogram at the bottom, you can see their different ages. So we have a range of age and the phenotypes associated right before birth through to adulthood. And then on each line, we've got the phenotypes and whether they are present or absent in the individuals in each group. These are sort of high-level groupings of phenotypes. I'll draw your attention to a couple of things here. Firstly, we've got neurodevelopment. So this is the darkest line that's present in almost all the individuals with this condition, present from about the age of 1 year, at the point where you can start to actually assess this. Similarly, short stature, the first line here after scan abnormalities, again, very prevalent across all the individuals who have this condition. But higher up in the graph, we have other phenotypes such as cardiac phenotypes, gastrointestinal phenotypes, or dental phenotypes, which are present either later or only in a small proportion of the individuals. So the syndrome itself is highly variable and presents quite differently in different individuals, different families. The other thing that's interesting in the DDD study about this particular syndrome is that although it is a classic de novo dominant condition primarily caused by de novo mutations not present in either parent, in DDD we see nearly 10% of cases are inherited from an apparently unaffected parent. And this is what brings me on to incomplete penetrance. To put simply, penetrance is the probability that an individual with a pathogenic genetic variant develops the specific disease. So in the picture you can see complete penetrance would be everybody with that variant develops that disease. And incomplete penetrance is simply where the genotype is no longer predictive of the phenotype, and some individuals with the genotype do not have the disease. We have a related but somewhat different concept of expressivity, which is the degree to which the trait expression differs, either quantitatively or qualitatively, between individuals. So it could be severity, or it could be the presence of individual phenotypes like in ANCHOR and D11. And you can see here, even for 100% penetrant phenotype, you can have variable expressivity. And then, of course, for something like ANCHOR D11, what we see is incomplete penetrance and variable expressivity. So a couple of just classical examples that hopefully you all know of penetrance. So a classic example to understand penetrance, I would say, is metabolic disorders. So PKU caused by autosomal recessive loss-of-function variants in the gene PAH. And this is, you would talk about it being a 100% genetic penetrant condition, but it does require dietary phenylalanine, and therefore it can be prevented by restricting the diet and avoiding phenylalanine. So this is a condition where we can talk about the environment affecting the penetrance of the genetic condition. Another classical example of variable expressivity and incomplete penetrance is FML1, which is associated with Fragile X syndrome, where repeat expansions of a CGG triplet repeat cause the condition, but the size of the repeat affects both the expressivity and the phenotype, but also the age of onset. So these are kind of classical examples from family-based clinical studies where we've seen incomplete penetrance and variable expressivity. And I think one of the things that we're starting to realize that there's a sort of unifying threshold model of disease where we can think about each gene having a certain expression level in a given tissue or an organ, and that expression level being dependent upon variants in that gene and a number of other variants, which I'll come on to. And perhaps in individuals with particular disease-causing variants, that threshold, the amount of product that they will produce from that gene could be a lot lower. And at some threshold, there's a detectable phenotype that we would then associate with a disease label. And obviously, if that threshold is higher, we would see potentially an autosomal dominant condition or a higher penetrant condition. And so the threshold model, thinking about the level of the product of a gene in a cell, is a useful model for thinking about penetrance and understanding it. We can also think about what actually modifies penetrance and expressivity, and I'll come back to this again later in the talk. The first is obviously different causal variants within the same gene, different variant location, different variant consequences, whether they're loss of function, whether they cause a dominant negative effect, and obviously the repeat expansion. So different causal variants in the same gene can potentially have different penetrance. This may seem obvious, but I think we need to move away from talking about penetrant genes and penetrant syndromes and towards thinking about the penetrance of individual variants or types of variants. We can then think about the level of gene expression, of which there's a number of different modifiers. So we can have different allelic expression, so expression of the two copies of the gene, different isoforms, so we've seen in other sessions different RNA isoforms produced from the same gene can result in very different products. So, for example, a loss-of-function variant, a stop-gain in one transcript could be intronic in another, and obviously the penetrance, therefore, and effect in different organs will be different. Cis and trans elements affecting the level of the gene expression, somatic mosaicism, where the individual doesn't necessarily have a causal variant in every single cell, which would obviously affect particularly the expressivity and potentially the penetrance. I think we also need to accept that there's a stochastic element to this. Gene expression varies during a cell and during development. Sometimes it'll be higher, sometimes lower, and that can have a major influence on the developmental trajectory of that cell. Whilst we like to hope to have all the answers in genetics, I think there will always be a stochastic element that we'll be able to model probabilistically, but we won't be able to have all the answers. And then finally, there are some global modifiers that will affect the penetrance of individual variants and individual disease-associated genes. So the polygenic risk score, for example, genetic compensation or genetic buffering by other genes in pathways that can compensate for the problematic gene. The efficiency of nonsense-mediated decay, so whether the protein product is made or whether the RNA is degraded. Obviously, the family history affects the penetrance significantly, and family history is an incredibly useful clinical tool for evaluating risk, and obviously is a whole mixture of genetic and environmental factors. And as I've already said, the age and the sex of an individual can affect the penetrance, as can the environment. So a whole range of modifiers can alter the penetrance of a particular variant for a particular condition. And really what we've started to think about increasingly is, if we see a pathogenic, or when we found a pathogenic variant in a patient or an affected family that causes a particular condition, traditionally these have been seen often as very highly penetrant. At the very least, the majority of individuals will be penetrant in that family. And in those cases, they have been selected for that reason. We found highly penetrant variants in a highly penetrant background with modifiers that allow that phenotype to be present, because by definition, that's how we've selected those individuals into that cohort. What happens if we now go to a different ascertainment context and look for the same sorts of variants? So similar variants associated with these sorts of developmental disorders in the general population, or at least populations that are recruited in a way that's agnostic to their family history or their symptoms associated with developmental disorders. And in this set, of course, you have a much lower prior probability that any individual has a particular monogenic cause or a particular developmental disorder, or even potentially a genetic condition. So we can use these two cohorts to compare and understand something about the penetrance and indeed the modifiers if indeed we do see pathogenic variants in both cases. So of course, you will, I'm sure, have heard about UK Biobank extensively already in this conference. I won't say too much other than it's a voluntary cohort of about 500,000 individuals in the UK, age 40 to 70 at recruitment, lots of genome data, quite extensive phenotype data. What I want to highlight in this talk is particularly the fact that they do have a strong ascertainment bias. They have a strong healthy volunteer effect. These are individuals who were sent an invitation in the post. Around about 5% accepted that invitation. And they are all over the age of 40 at the point of recruitment. This means, by definition, none of them had a terminal disease before the age of 40. That's obvious, but worth highlighting. All of them were capable of giving consent for this project and had enough time and resources to participate. They have a survivor bias, so if you look in the graph here, they are literally dying at a slower rate than the average UK population, and they are healthier, wealthier, and more educated than the average of the UK. So they have an extreme ascertainment bias, perhaps almost opposite to what you might see in the clinic, and you would certainly expect the cohort to be depleted of severe pediatric developmental disorders. So we and others have asked the question, what sorts of developmental disorder phenotypes can we see in UK Biobank? And there's really a mixture from self-reported questionnaire data and hospital ICD-10 codes, as well as GP data. You can look at medical diagnoses relating to child or adult onset developmental disorders, so epilepsies, intellectual disability, developmental disorder, congenital malformations, and schizophrenia and bipolar in adults. There's a range of cognitive traits, so everyone who was enrolled in Biobank was asked to do a number of quizzes as well as fill in questionnaires around fluid intelligence. They were able to look at reaction times, their ability to match pairs in a particular test. They evaluated the years in education and so forth. We have data on their socioeconomic background, data on the number of children they have, and various physical traits, all of which we would expect to be correlated with genotypes that would cause developmental disorders. So how do we find those? There's really a mixture of different things informed by family-based studies. Of course, classically, many developmental disorders have been caused by very large copy number changes, so in the extreme chromosomal aneuploidies, such as X chromosome, monosomy, or trisomy 21, Down syndrome, as well as a number of large multigenic CMVs that are associated with syndromes. Increasingly, in the era of whole genome sequencing in particular, we know lots of small single nucleotide variants and indels associated with these disorders as well, which we can find from the exome and genome sequencing in biobank. We've particularly looked at genes that have been clinically curated to be associated with severe developmental disorders in the clinic. So I'm going to talk about this DDG2P list a number of times. This has got more than 2,000 genes where variants have been shown to cause developmental disorders. It's one of the founders of the Gene Curation Coalition. And so we have a set of genes that are very well known and very definitively associated with developmental disorders, linked with their inheritance pattern and also the mechanism of disease. In biobank, we can then pull out predicted loss of function variants, and we use just the variant effect predictor for this, as well as the lofty high confidence variants, which are more likely at least to cause loss of function. We can look at missense variants in these genes and whether they're likely to be deleterious or not, whether they've been marked as pathogenic or not in ClinVar before, and of course, like many people, we can use synonymous variants as a control. So what do we see potentially with the starting point that we're maybe not expecting to see very much in biobank at all? So starting with the biggest variants first, this is a really nice piece of work led by some of my colleagues, Anna Murray in Exeter, looking at X chromosome aneuploidy in the UK biobank. And actually there are quite a few hundred individuals who have Turner syndrome and XXX syndrome in biobank. The majority are mosaic, but many you would encounter clinically are also mosaic. And you can see in this graph of height that the height of individuals actually overlaps extremely well with the background population. So in the red, we have non-mosaic Turner syndrome. In the orange, we have the mosaic Turner, and in green, XXX. And in the background, you can see the background histogram. And this is true of a number of the traits that, although there are individuals at the extreme ends of this, by and large, they have phenotypes that are consistent with the clinical phenotypes, but they are milder than we would see in the clinic. And this obviously has implications for how we interpret particularly mosaic X chromosome aneuploidy. Moving down a step to these multigenic CMVs, or CMVs that have been associated with syndromes, this is a really nice piece of work from George Kirov's lab in part of the UK, looking at a set of well-known syndromic CMVs and their penetrance based on presence in cases versus control, and then a sort of average score of cognitive traits and indeed the number of children they have from UK Biobank. And what they hypothesized and indeed saw is that the penetrance is higher where the effect size is higher. So a bigger effect means you're more likely to see a penetrant phenotype. So then if we go down to the smallest unit, just looking at a single gene potentially, I'm coming back to this ANCAR D11 that was associated with KBG syndrome that I mentioned at the beginning. In this gene, you can actually see burden testing of carriers of loss of function variants in a UK biobank. There are 18 and about 400,000 exomes at this point. And you can see there's a statistical enrichment in these individuals where they are shorter than average, as we saw with the individuals with diagnosed KBG syndrome. And they also have particular disorders associated with school skills. Most genes, it has to be said, don't have enough variants in them to do individual burden testing. So what we started to do is think about how can we aggregate variants together across multiple genes. And obviously you can pick various different subsets of genes to do this. So we use the DDG2P set, and the 4 points on these graphs are different, increasingly stringent subsets. So the first one is the top 25 genes, where we have more than 30 de novo diagnoses in the DDD study. So these are really, really certain highly penetrant monogenic disorders. The next set is 125, which are genome-wide significant in a large analysis of 31,000 trios and de novo mutations. The next one along is 325 genes, and these are where we know the mechanism is loss of function. So we're expecting to see loss of function variants causing these individual genes. And the final one, we have about 600 genes, which are just dominant developmental disorders. And you can see there's an association in UK Biobank with the diagnosis of developmental disorder, with the Townsend deprivation index. So essentially these individuals have a lower socioeconomic status. Their fluid intelligence is lower and their height is shorter, but that effect is attenuated as we increase the number of genes, potentially because we're including variants that aren't causal in those genes, and potentially because those genes have smaller effects associated with them. So if we look at just the 600 genes, this is the biggest gene set there, there are rare damaging variants in the UK Biobank across thousands of individuals in these genes, which is a little bit of a surprise initially. And these are associated with milder, perhaps subclinical phenotypes in many cases, but they are developmental disorder-related phenotypes. And again, the variants with larger predicted effects are associated with larger phenotypic effects. So that's just a summary here: decreased intelligence, reduced memory, shorter stature, fewer children, fewer years in education, less likely to have degrees, less likely to be employed, lower income, higher BMI, more likely to be unable to work, higher deprivation, and indeed slightly more likely to have a diagnosis as well. So I'll just show you a little bit of data here, and all the graphs I'm going to show you here, there's a clear trend. So the first in each of these is a large CMV multigenic copy number deletions, followed by multigenic duplications, followed by predicted loss of function variants across the 600 developmental disorder genes, followed by missense, like plausibly deleterious missense variants, followed by synonymous. And you can see for both of the diagnostic categories, there's a clear trend where there's an increased odds ratio with a larger effect variant. We see a similar thing in traits related to intelligence and education, so a trend from the largest effect variants down to synonymous, which are our controls. And similarly for the socioeconomic traits. So what we think is happening here is, as I alluded to earlier, a difference in the ascertainment bias between clinical cohorts and population cohorts. In the clinical cohorts, it's very much a phenotype first. An individual has a developmental phenotype, which is the reason they're being offered genome sequencing, and then we are biasing towards more penetrant variants, but also a more penetrant genetic and indeed environmental background. In population cohorts, we're doing a genotype first, essentially, and biasing potentially towards lower penetrant variants and indeed a lower penetrant background. Therefore, potentially, the general population may well sit somewhere in between the two. But I think the two cohorts potentially, therefore, give us an upper and a lower bound of penetrance. Why is that likely to be true? So I've tried to divide this really into a couple of different reasons for the differences between different studies. So the first is around molecular mechanisms that potentially different variants in the same gene or biological rescue of individual variants. So although in some cases there are identical variants, we still might see different penetrance. I'll then look a little bit at modifiers, particularly genetic modifiers, and then finally touch on, unfortunately, errors, which can also creep into these studies. So the first one, if we just think about local, global, and stochastic modifiers. You have the causal variant in your gene of interest, which I think in these developmental disorders, the monogenic ones I'm describing, there's likely still to be a primary variant that is perhaps the largest effect. And of course, that can be affected by, for example, cis-regulatory variants that are in the promoter or the enhancer of that gene that might increase the dosage of the wild-type haplotype. It can be affected by the rate of nonsense-mediated decay or the efficiency in particular cells, and a number of global modifiers. So firstly, just looking at the position of the variant. So we really focused here on loss-of-function variants. And as we heard on the first day from Caitlin, not all predicted loss-of-function variants truly do cause loss of function. So we tried to look at whether we could discern something from clustering of loss-of-function variants. So people have looked before at missense constraint and clusters of missenses. We tried to look at where are loss of functions in UK Biobank versus clinical cohorts, and can we learn something about loss of function constraint from that? And really what we see is there are sets of genes where there's much lower constraint in the starts and ends of genes, and in particular exons, which are likely rescuing what appear to be pathogenic variants through a variety of mechanisms, such as nonsense-mediated decay or alternative transcription. And, for example, translation reinitiation. So this is the example I'll give here of GATA6, where loss-of-function variants are associated with cardiac condition and also pancreatic agenesis. And we see, by and large, benign truncating variants in blue here at the start of the protein, which are primarily where we see biobank variants, and indeed variants in NOMAD, versus similar sets of loss-of-function variants that are pathogenic in CLMVAR and other databases. And really what we think is happening here is that these early variants are being rescued by an alternative start, an AUG, which you can see is just positioned before the start of the structure of the protein domain that conducts the function of this gene. And so these early variants, translation reinitiation occurs on the mRNA molecule and rescues the protein so that although you have a shortened protein, it's still able to do most of its function. And actually, we're able to draw some sort of broad conclusions from this work. There's three sets of genes, essentially, is what we're suggesting here. There's a set of genes where we just don't have enough predicted loss-of-function variants to cluster, so in places like UK Biobank. So, for example, STXBP1 that we heard about the other day, there are hundreds of loss-of-function variants in ClinVar and other databases, and almost none in UK Biobank. It's very likely that these really are fully penetrant conditions, where a loss-of-function variant is in that gene, the individual will get the condition. The next set is where you get distinct clusters of loss-of-function variants, much like GATA6, where there are differences in the clustering of variants in population versus clinical cohorts. And there we can start to postulate molecular rescue mechanisms that are different within the gene itself in those particular cases, that explain the difference between pathogenic or severe variants and benign or mild variants. And then there's a puzzling set, which is unfortunately the largest piece of the pie, where we don't see any clustering and we don't see any difference between where the loss-of-function variants are in the clinical cohorts versus the population. So something else must be explaining cases of incomplete penetrance or indeed milder phenotypes here. So one hypothesized, an obvious answer here, particularly for the haploinsufficient genes, is that you have essentially haploinsufficiency rescue on the other haplotype. So thinking back to our anchorin D11 case example, where you've got an unaffected carrier parent who carries the large effect loss-of-function variant, if they have a variant in the promoter region, let's say, or an enhancer of that gene, which increases its expression on the other haplotype, then thinking about the threshold level, it could be above the threshold that is required for the protein to do its job, whilst the child doesn't inherit that compensating variant and therefore is below the threshold, and you'll see the disease. And there's certainly some really nice evidence from the Castell paper in Nature looking statistically at this and a reduction in these sorts of variants in the population. But there can also just be additional variants. They don't necessarily have to be only cis to the gene itself. They could be anywhere in the genome. There could be multiple large effect variants or small effect variants that we can aggregate together in a polygenic risk score. And these can alter the penetrance of the primary large effect variant. So this is something we've looked at very recently. And what I'm going to show you here is the educational attainment polygenic risk score, just broken down into deciles in UK Biobank versus the effect on fluid intelligence on the y-axis. And the dotted line is for most of Biobank, for individuals who have no large effect variants in these DDG2P genes. But then we looked at individuals who carried one or more variants in different genes. And you can see that the effect on their intelligence, it's an increasing effect, so their intelligence is lower the more rare damaging variants they have, but the trend is the same in terms of the polygenic risk score, so that if they're at the low end of the risk score, but the high end of the variant burden, they could actually have the same effect. And we see this across a number of different traits, education, income, and Townsend deprivation index. We hypothesized that individuals in UK Biobank, because they are so overly educated and healthy and wealthy and so forth, must have something that's sort of protecting them. So if they're carrying these deleterious variants, why aren't they having a bigger effect? And indeed, you do see a higher educational attainment polygenic risk score in these DD variant carriers than you do in the rest of UK Biobank, suggesting it's compensating to some extent for that problem. Conversely, those who actually have a diagnosis in Biobank, they've actually got a lower, substantially lower polygenic risk score, which again suggests potentially in order to actually get a diagnosis and still be in this cohort, it's very likely potentially that their causal variant is not having quite such a big effect, and therefore the polygenic risk score needs to be that much lower to actually push them over that clinical threshold whereby they have a diagnosis. And the last thing we looked at here was what we called phenotypic deviators. So, for example, individuals who have a very low fluid intelligence score, the very bottom of the scoring range, but a really high polygenic educational attainment score. And can the rare variants explain why we see these individuals who appear to have the opposing phenotype versus that predicted from the polygenic risk score? And indeed, you do see an enrichment in these individuals for these rare damaging developmental disorder variants where they're much more likely to have more severe and higher effect variants than others in biobank. So their polygenic risk score is partially compensating for the DD variants, and the opposite can occur as well, so that the two are additive, essentially. And this really supports, essentially, this sort of liability threshold model, where numerous factors can potentially contribute to a clinical presentation. So for some genes and variants, they might be like this first example here, perhaps STXBP1 is like this, where every individual is in this red box. If you have a loss of function variant in this gene, there will always be above the threshold for a clinical presentation. But many other conditions might be more like these other two, where the level of gene expression plus a number of modifiers will affect whether an individual with this large effect red variant here are above the threshold for clinical presentation. And we don't often know what these modifiers are, and we don't really know how to look for them. We might know statistically broadly what they are, but for an individual, we don't necessarily know, which makes it really hard to interpret just this individual large pathogenic variant in the absence of a phenotype or a family history. So the last thing I wanted just to touch on is some of the problems that can occur in these penetrance studies. So I've talked about a lot of very large data sets that we're using to evaluate these individual variants and genes and phenotypes, and a lot of things can creep in when you do these large-scale studies. So one is the inclusion of variants with high allele frequency. So I think historically there have been a number of variants associated with monogenic conditions and marked as pathogenic potentially, but we now know because of population data they're actually far more frequent than is possible for a causal variant. And so including these sorts of variants in any study of penetrance is necessarily going to bias your penetrance estimates down because they are essentially not causal. There are, of course, potentially errors in genotype calling. The very simplest would be a mosaic variant rather than germline. So although it's present in the data, it's actually not present in the majority of an individual's cells, so can't be contributing to the penetrance of a neurological phenotype, let's say, if it's only present in their blood. But there are other sorts of errors that can occur in genotype calling, complex repetitive reasons, for example. Indels are obviously sometimes more challenging. So in an ideal world, one would validate all these variants. Of course, you can't do that across thousands of variants, across hundreds of genes, in hundreds of thousands of individuals. But ideally, would look at least at IGV plots and evaluate the evidence with which the variant has been called. One big problem is refuted gene-disease associations. The literature is increasingly, I'm pleased to say, filled with refutations of previous gene disease associations. So where we once thought variants in a particular gene caused a particular phenotype, we now know that they don't. And that was, again, really, many of the papers date back to the era before we had population data. And there are quite a few genes now where the gene disease association has now been refuted. So again, including those in any sort of data set which you look at will be problematic. So it means you really need to keep on top of the literature as to which genes are associated with which phenotypes. Of course, we all know that databases of variants are incredibly useful, but they're not perfect. So pathogenicity assertions can change over time. They can be revised. They can be wrong. And therefore, it's obviously challenging to make sure you really do just include truly pathogenic variants in these studies. As an aside, of course, it's quite difficult to think about what we even mean by pathogenicity. If a variant is extremely low penetrance, is it truly pathogenic, or is a different term more appropriate? And I think that's something we're really going to have to grapple with as we see more and more apparently pathogenic variants that have penetrances of maybe less than 10% in general population cohorts. Do we still call them pathogenic at all? You can have problems with variant annotation. So we've already heard about loss of function variants that are annotated as such. They're protein truncating, let's say, but they don't actually result in a loss of function. So the effect prediction of a variant is just that. It's just a prediction. And obviously it could be a prediction that's not necessarily in the correct transcript for the correct phenotype of interest. The inappropriate mode of inheritance will also affect penetrance, so particularly including autosomal recessive conditions in a study of heterozygotes. Unless you're actually interested in the phenotype associated with carriers, that would obviously influence your penetrance. The incorrect mechanism of disease is really important. DDG2P and the G2P database generally is unusual in that it has mechanism of disease stored for every single gene disease association, and therefore you can focus on just those where loss of function variants cause a particular disease, separately, for example, from gain of function or dominant negative. And obviously, looking at the penetrance of loss of function variants for a condition which is caused by gain of function is not going to be very helpful. Phenotype definition can be extremely challenging. I think genetics has improved incredibly, and the standard of genotype data is now incredibly high, but not necessarily true of all phenotype data, and there are different ways of thinking about how we define different phenotypes. Obviously, the age of an individual is very relevant, perhaps less so for a congenital developmental disorder, which should be present from very early on, but some other conditions are late onset, and therefore you can't evaluate them until the age is appropriate. And finally, there are different ways of calculating penetrance, some of which perhaps are more appropriate than others. So I'll just show you one brief bit of data which is IMPRESS, so I'd rather not have any photos, but just looking at one set of variants that was published associated with diabetes, where all of these errors had occurred to some extent. And I do think all of these errors are actually pretty easy to make unless you go in and hand curate individually every single variant and your phenotype. And on the first graph there, we can see just individual variants, and what we're looking at here is difference in absolute risk of diabetes between carriers of variants and non-carriers. So some of them actually have a lower risk simply because they're just in the population and they don't have diabetes. It's not that it's protective. And there's a whole range, and you can see they're actually extremely common. But the problem is that the penetrance apparently is 0. And these are supposed to be pathogenic variants in monogenic disease genes. When you curate these variants and these genes, based on the 10 points I had earlier, you'll see you have much more sparse data, you have fewer carriers, and indeed the penetrance now is much higher, at around sort of 50 to 60%. So curation of your genes and variants makes a big difference. And ideally, you might actually look at something like lifetime penetrance, where you look at the penetrance of a particular condition at different ages and in different cohorts. You can see in green here, this is a cohort of monogenic individuals who have monogenic diabetes, so probans. So by definition, the penetrance there is 100%. Then in black, we have their family members, so they get diabetes slower, but they do all eventually get it if they carry the variant. Then we have a medical scenario, Geisinger Medical in the US in red, and then UK Biobank in blue. And you can see that individuals get the disease later, and some individuals never get the disease, despite having the same set of curated variants. And so what we see, again, is this sort of decreasing penetrance with a decreasing prior probability of disease. So just my last slide and take-home messages. Firstly, incomplete penetrance and variable expressivity are widespread. They're not universal, but they are very widespread. And this allows apparently pathogenic variants to be present in subclinical populations. Developmental disorders is what I've mostly talked about today, but it's one of many examples. I think we will see this more and more as we evaluate more conditions. And this obviously has major implications for genomic screening and incidental findings. We have an opposing bias between clinical and population cohorts, which can be very advantageous when you're thinking about what are the upper and lower bounds for penetrance. And then there's a range of modifiers that can explain that. And I think a lot of research over the next 5 years in this area will really be focusing on trying to understand those modifiers better. And finally, as is always the case, curation of your data is really, really important, and it makes a big difference to how you estimate penetrance, and particularly if you're doing these sort of grouped variant analyses, which we will see more and more, errors can really result in an underestimate. So finally, just a brief thanks particularly to Becca Kingdom, a PhD student of mine, and Rob Beaumont, a postdoc, Mike Whedon, a professor of bioinformatics in the department, the whole of the EXA team, and all of you for listening. Thank you very much.
Excellent. So we're now going to open the floor for questions for Caroline. Please come up to the mics while we're waiting for people. So do you think there is one estimate of penetrance, and could we get it if we sequenced, for example, a birth cohort and followed them through life? Or, I mean, I liked that last plot there with the decreasing penetrance with decreasing prior.
I don't think there is one penetrance. We've had many discussions about this and maybe renaming the term, particularly to label it with the population from which it's derived. I think a birth cohort might give you the most useful penetrance, in a sense, if you're just going to evaluate an individual who walks in off the street. But there isn't one penetrance. I think it depends on the modifiers. So you have a clinically ascertained high family risk penetrance, and you have a biobank ascertained, and all the ranges in between. So unfortunately, I don't think we're ever going to get one number.
Bad news for us. Over here.
Hi, Bruce Galb, New York. Gorgeous talk, Carolyn. I just wondered what your vision is for how we're going to deal with what seems almost an insurmountable power problem, right? So we have a lot of genes, and we have to get to the variant level. We don't have polygenic risk scores for a lot. I mean, we have it for educational attainment and height, but we also know for many things we don't. And by the way, those obviously don't work in many populations, which is problematic. And then if you start to bring in the gene by environment piece, which we have struggled always to power anyway, how do you see us getting to the kind of accuracy that you're speaking to?
Yeah, it's a great question, and obviously is a problem. I guess there's two answers. One is more is better. So I think sequencing more cohorts, more diversity, obviously, geographic diversity, more clinical cohorts, getting really good phenotypes alongside really good genotypes, potentially functional data. So I do think some of that is the answer. That said, you could sequence everyone and you still wouldn't have enough power to find really rare modifiers. So I think that probably has to be coupled with individual studies, functional studies following up individuals where we see particular things, maybe looking at CRISPR screens, et cetera, where we can provide functional data. So I think probably the two have to come hand in hand.
So Caroline, you said that we have to create every variant. There was a beautiful example in your, I think it was GATA6. You know, there is one non-innocuous, so probably loss of function, but it was before the second ATG. So what's going on there? Did you look at it? I'm sure you did.
I did, yes, well spotted. I guess, there's two explanations. One is that it's wrong, and that it was called pathogenic in an individual because loss of function variants in that gene cause a condition. There is no phenotype associated with that variant in ClinVar, so I don't know. The other possibility is that there are exceptions to the rule, and this individual has other compensating factors, which mean that, in actual fact, in that particular case, or other problematic variants, perhaps they have another loss of function variant later on that we don't know about. So unfortunately, I don't know, and I think we have to accept that any rule won't be 100%.
Right. So it seems like we still have time for a question, but it seems like you're talking about moving into more of a probabilistic framework about if we take in your whole genome at the same time and any kind of risk factors that might be floating around your family, that gives us a better sense of whether or not this particular variant is going to actually express / give you the phenotype that we think is associated with it.
I think that would be ideal, a beautiful Bayesian framework which takes into account the population from which the individual has come from, why they were being sequenced in the first place. It takes into account things like constraint, evolutionary as well as population constraint, takes into account information about the protein structure and the function and its expression in different tissues. This is obviously a super-duper piece of Bayesian high-tech prediction, but I do think that probably is the way to go so that we can just have a probabilistic framework for suggesting whether a variant is causal in that particular case or not. And indeed, if it's a VUS, what we could do to try and establish whether it actually is causal or not.
That's where it exactly feels like it's quite useful. Excellent.
Thank you.
Thank you.
Our second speaker will touch about resilience. So instead of aggravating a phenotype, he will also say about protecting about a phenotype. And so Thomas Beaugeron is coming from the Institut Curie in Paris, and he will speak about phenotypic